delimitation. Yes. So Shahan Derkarabetian will be talking about the challenge of delimiting cryptic species and the supervised machine learning solution. Are we there? Is the sharing working?
Can you hear me? Can you see me?
Yes, very good.
We can. Okay. Shahan, you're in presenter mode. You need to switch it.
All right, one more time. How about now?
That's good. And we can hear you. Perfect.
Okay. I can't believe I have to go after Caitlin. All right. So, hello, my name is Shahan Derkarabetian, and I think about Apiliones a lot. However, I have to admit, over the past couple of years, I've been thinking about something other than Apiliones, and that is species delimitation. And I've been thinking about this because of what I see in the field. We obviously want better and better computational tools, but I feel that it's increasingly at the expense or to the exclusion of considering the biology and natural history of the organisms we study. So the purpose of this project and presentation is to push back against that and show that we can have both. We can have a computational tool that is ideally suited for species delimitation and combine that with our knowledge of the biology of the organism all in a single analysis. And I'm going to show that by showing you how I have delimited cryptic species using genetic data. So delimiting species can be difficult for many reasons. One of those reasons is because speciation is a continuous process, and it can be difficult to know when and where to draw the line. Another reason is due to the diversity of the taxa we are trying to delimit, particularly with genetic data. And this is because different organismal types have different underlying genetic patterns associated with populations and species. So for example, a species of albatross will have different underlying genetic patterns compared to a species of montane salamander. For us arachnologists, we can think of a species of Habernatus will be different from a species of Hypochilus. So how we delimit species in practice is dependent on the biology of the organisms we study. The organisms I study are Apiliones, specifically low dispersal Apiliones. Now, low dispersal Apiliones come with a particular set of biological characteristics. They tend to undergo non-ecological speciation, and this includes many different things. First, nested allopatry. This just means distinct genetic lineages tend to rarely, if ever, occupy the same geographic space. They also show niche and morphological conservatism across species boundaries. They tend to have low dispersal ability and high ecological constraints, which both lead to inherently high levels of population genetic structure within a species. Now, all of these together lead to, have the potential to lead to cryptic speciation. These characteristics are, of course, not just limited to Apiliones or arachnids. They're found across the diversity of the tree of life. However, they are most problematic in taxa like Apiliones. So in taxa like this, morphology and ecology will lump species level diversity. We cannot use characteristics like reproductive isolation. So we have to rely on genetic data. In the case of cryptic species, only genetic data. However, there's a problem. All of the genetic species delimitation analyses tend to oversplit and instead recognize population genetic structure. This is something that has been shown across a wide range of empirical studies, from arthropods to vertebrates, I forget what they're called, and plants. So the question is, how do we delimit species or cryptic species using genetic data? So my solution to this problem, after thinking about this for 2 years, is a supervised machine learning approach. Supervised machine learning approaches, when applied to this problem, are classification tools. We use a labeled training data set with known information to inform an unknown testing data set. And I'm going to show how this works, focusing on this awesome species called Theromaster bruneus. This species is distributed throughout the southern Appalachian Mountains and surrounding region. You can find it associated with leaf litter, underneath woody debris, and rocks. This species has an abnormally large distribution for a northern temperate laniatory species. This is particularly interesting considering the geographic location, where the vast majority of taxa in this region show high numbers of species. So having one species across that broad distribution just doesn't make sense. So we did an integrative taxonomic revision, well, technically of the genus, but pretty much of this one species. So starting with morphology, as always. Across that geographic distribution, we see high morphological conservatism. There is no variation except for this one individual, this one female we found, where we would call this a morphospecies, a distinct species based on pigmentation patterns, eye mound shape, spines, body shape. It's obviously something new. So taking a little bit deeper, looking at sexually dimorphic chelicerae and pedipalps, again, there's no variation across the distribution. What about male genitalia? No variation. So this is obviously a very highly morphologically conserved group. So now getting into genetic data, I'm going to start with DD-RAD because this was the most well sampled. What we see are deep genetic divergences between some lineages we have named and colored based on geographic location. And these clades are, for the most part, geographically exclusive. Going a bit deeper, we see high levels of population genetic structure, even within the most derived lineages. Comparing across genetic data sets, here we have our UCE data. The topologies are highly congruent, except for this purple clade, but that's okay. Looking at Co1, it's a bit messier. There's some congruence in the early diverging lineages here, the black and green. However, once we get to this node here, it all breaks down. There's no cohesiveness anymore. So summing all of this up, that morphospecies I mentioned earlier is this single individual here. And then the rest of this genetic diversity is represented by that highly conserved morphology. So given what we know about the biology and the geographic region, the question is, how many cryptic species are there? So we did a lot of species delimitation analyses, and it wasn't friendly to us. No data set or analysis was congruent with there was no congruence, basically. So I'll say at this point, as taxonomists, we had a gut feeling or an initial idea based on tree topology that there were 3 species. One of the analyses reflected that. However, there are some technical difficulties associated with that analysis I don't have time to get into. So we sat on this for a very long time. We didn't know what to do. Well, we being me. We didn't know what the species were. We didn't know what to do. And then this project kind of just sat there for a long time. Eventually, I said, okay, I want to try a supervised machine learning approach. A few years ago, this program called Clades came out. This is a support vector machine learning framework. It uses a 2-species model to classify samples as either the same or different species. Now, with this program, they introduced a training data set for this. And I call it a general training data set because it's from simulated data based on population parameters representing a broad range of plant and animal taxa, so very general. They applied it to multiple empirical systems, and they showed that it worked across those systems. It correctly estimated the number of species. That's great. That's amazing. So what happens when we apply it to Theromaster bruneus? It fails. It says every single population, in this case, every single individual is a distinct species, which we know is not true. So now the question is, why does this general training data set fail? And more broadly, why do all genetic analyses fail in taxa like this? And I think about it this way. Imagine an n-dimensional biological characteristic space. All of the biological and ecological characteristics that go into explaining the genetic structure within a species. You can imagine a general training data set, or even any fixed model that we use, may occupy this much space. It works in this space here. However, somebody's particular study system may only occupy this much space, and somebody else's particular study system may occupy this much space. If you're like me, your study system does not exist in that space at all. And that is why these analyses fail. So the power of a supervised machine learning approach is in making custom training data sets for each of these individual study systems. So before I show you where I got my custom training data set for genetic data, I'm going to do a little thought experiment with you to show you why this is important. So we're all experts on some arachnid lineage or multiple arachnid lineages. Now imagine a new species is described, like this beautiful species here, Synonychia martinsi from a cave in China. This new species gets described and you're excited about it. But all we know about it and all we know about the vast majority of species we describe is what it looks like and where it's found. And you tell your friend about this, right? Hey, check out this new species. If your friend is a good friend, they'll ask you a question about it, like, what does it eat? So the 100% honest answer to this question is, I don't know, period, end of sentence. But we don't answer that way. We give them some information. So for example, here's a table from 2007 that lists all of the feeding observations we knew about Apiliones. This we can consider a general training data set. If we answered this question using this data set, we would say, I don't know what it eats, but it eats everything, because Apiliones as a whole eat everything. But that's not true for this species, because you can't find everything in a single cave in China. So it makes more sense, thank you, it makes more sense to focus on a particular group, the most relevant observations to this particular species. And when we do that, this data set becomes a custom training data set. This only includes Laniatorian taxa from northern temperate caves. And when we use this custom data set to answer this question, we say, I don't know specifically, but it probably eats columbolins and small beetles. That's a much better answer. Your friend is happy for it. So the question now is, where did I get the training data set from? A couple of years ago, we published this paper demonstrating unsupervised machine learning and species delimitation. Let's completely ignore the machine learning part of this, because at the core of this paper was an integrative taxonomic revision of the genus Metanonicus. This genus is distributed throughout the beautiful Pacific Northwest. You can find it in leaf litter associated with woody debris and underneath rocks. We did a bunch of species delimitation analyses in this system, and species delimitation was very easy. There was high congruence across data and analysis types. So I have 100% confidence in these species, and that's important. So I used Metanonicus as the custom training data set for Theromaster bruneus. And this works because they have the same biological and ecological characteristics, the same microhabitat preference, and the same modes of speciation. Therefore, they should have very, very similar underlying genetic patterns associated with populations and species. So I now have a custom training data set for low dispersal Apiliones. When I apply it to Theromaster, here's what happens. I get 2 species. That's amazing. I was so happy when I saw this, because it's a reasonable answer. And that's all I ever wanted. So when you combine these 2 cryptic species with this third morpho species, we get our 3 species, which agrees with our initial gut feeling, our initial ideas, before we ever did any of these species delimitation analyses. So this approach is universally applicable, meaning you can make custom training data sets specific to any organismal type and data type. So for example, I just used UCE data because they have specific characteristics. So this approach, I rely on the flexibility of machine learning to accommodate biological variability. And most importantly, this approach combines the objectivity desired from computational species delimitation with organismal expertise based on our years of experience and our knowledge of the natural history of the taxon. So obviously the next step is to delimit all the cryptic species. I'm excited to use this. I'm excited I found something that works. And I'm excited I can stop thinking about species delimitation so much and actually get back to, describing these species. So I'm also more excited about other people trying out this approach. And if you are interested in trying out this approach with your study system, I'm looking at you, my Gallimorph biologists. I'd be happy to show you how this works. So don't hesitate to ask. And with that, I'll thank you for your time and answer any questions, of course. Thank you.
Okay, unmute. Thank you so much, Shahan. Awesome talk. Yeah, we do have time for a couple of questions. And we have already one in here. Oh, yes. Okay, so on the number of species test, could you get into it for us a bit. Why the two likelihood spikes in number of predicted clusters at n equals 2 and n equals 7?
Yeah, so
with reference to your technical difficulties.
Yeah, I can see the question here.
Okay.
So that peak at 3 species, I don't really have an explanation for it. It happened in both the DDRAT dataset and the UCE dataset. I don't know. I think it's just long branches. You know, most datasets I work with, when I try the SNAP base factor delimitation, I don't see that. I just see a constantly increasing line with increasing species. So that is unusual. So in one of the data sets, it peaked at 3, and then it kind of increased and then stopped. So 3 was the highest. But in the UCE data set, it peaked at 3, but then continued increasing until the highest split, which was even more likely than the 3. And that's the technical difficulties, because all of these approaches are just dimensionality reduction techniques. And if I didn't test k equals 3, I would have never seen that. So it's all dimensionality reduction.
Thank you. We have to move on.